Hello, students. Hello. Good afternoon, students. Good afternoon, or rather, good evening, all of you students. Welcome back to Vedanta Science Olympiad. This is Basraj, sir, your biology master teacher, students. Now, I hope I'm audible and visible to all of you students. I hope I'm audible and visible to each and every one of you. Perfect, students. Perfect. Now, हमने students पिछले class में we finished something called as different types of tissues based on their ability to divide. For example, we finished something called as meristematic tissue as well as permanent tissue. Can anyone tell me, students, in the class right now? Anyone in the class tell me, students, meristematic tissues. Meristematic can be divided into two categories based on the origin. Anyone? Hello. Based on origin, we have two types of meristematic tissue. That is primary tissue as well as secondary tissue. Now, what about based on location? Based on location, we learned that there are three categories. First one is called as apical or SAM, root apical meristem and shoot apical meristem. Then there is intercalary meristem, and then there is lateral meristem. Three different types of meristematic tissues based on the location. Then we learned about permanent tissue. In permanent tissue, we learned about something called as your parenchyma. Then we learned about collenchyma, and then we learned about sclerenchyma. Right? Three different types of permanent tissues. Now they were simple permanent. Now can anyone tell me which are the two complex permanent tissue, students? Anyone in the class? Anyone in the class who can tell me what are the two different examples of complex permanent tissues? One is xylem, one is phloem. Xylem has different types of categories. There is tracheids, vessels, xylem parenchyma, and xylem fiber. Similarly, in the phloem also, we have sieve, companion, and then we have the parenchyma and the fibers. So, this basic difference which all of you should know before you attend today's class, because all of that is required to understand today's class, students. Okay. Now, if you look at the diagram here, this diagram is the typical sectioning of a leaf, or it could be any type of stem also, or a root also. A root, stem, or a leaf. In the case of monocot, dicot, any one of them, there is a typical pattern. What is the typical pattern, students? The typical pattern is that the entire thing is divided into three different parts. First of all, there is epidermis. Now, what is epidermis, students? Epidermis is generally the outer layer of a stem, outer layer of a leaf, outer layer of a root. फर्स्ट वन एपिडर्मिस. उसके बाद जो नीचे की तरफ जब जो कुछ भी है ना, that becomes the ground tissue, also called as the cortex. Also called as the cortex. Now, after cortex, what do you have? After the middle part of cortex, at the bottom, we have the vascular tissues. Now, what do you mean by vascular tissues? In vascular tissues, we have a special type of arrangement. Arrangement of vascular tissues. That is how the xylem and phloem are arranged. Now here we will learn something called as your radial, bicollateral, collateral, open and close in today's class. Okay. So before we go to the ground tissue and vascular tissue, let's quickly understand the epidermal part, students. What do you find on the epidermis? Now can anyone tell me, students, have you ever tried touching a tomato plant? कभी देखा है स्टूडेंट्स? कभी देखा है अपने लाइफ में एक टोमेटो प्लांट को? स्टूडेंट्स कभी देखा है क्या? कभी भी लाइफ में एक टोमेटो प्लांट को देखा है? If you have seen a tomato plant, on the surface of the tomato plant, on the leaf, on the stem, right, you will notice a small hair-like structure. Yes or no? Like if this is a leaf of a tomato plant or a stem of a tomato plant, you will notice a minute, small structures like this on the stem and on the leaf. Now we will be studying about what is that. Now after that, all of you might be aware that especially in the case of roots, this is the part called as, students. If this is a root right now, in this root there are three regions: meristematic, elongation, and this is called as maturation part. Now can anyone tell me, students, in this region of maturation, what do you find? In this region of maturation, you will find something called as root hair. Root hair can be observed here. So in the epidermis part, we will be learning about this trichome and root hair in the first part right now. Okay. So here is the information given for your students. for the epidermal appendages. See, there is something called as root hair. Root hair is there. So if this is the entire root, मेरिस्टेमेट रीजन, रीजन ऑफ एलोंगेशन, और उसके बाद आता है रीजन ऑफ मैच्योरेशन. इस रीजन ऑफ मैच्योरेशन के अंदर you will find on the outer layer of the region of maturation, you will notice a fine hair like structure called as root hair. It is exogenous in origin, it is unicellular, it helps in increasing the surface area. That you need to know about the root hair. Now uske baad what else you find on a plant? Now when you talk about stem or a leaf, remember I told you tomato one. In the case of tomato, you find something called as trichomes. You find something called as trichomes. Trichomes are very important. Trichomes are found on the stem or leaf, right? They are exogenous in origin. The meaning of exogenous is that they are present directly on the epidermal part. They are arising from epidermal part. Now when I say root hair is also exogenous, it is also coming from outside. Okay, so both are exogenous in origin. Okay, they can be uni or multicellular. They help in control the water loss. That basically all of these small hair like structure on tomato plant, they help to reduce the water loss. Reduce the water loss. They can be soft or hard, or septate or secretory in nature. So this is what you need to know about the epidermal appendages. The meaning of appendage is the outgrowth here. Outgrowth which we see here. Okay, so on the epidermis, all of these, both of these are called as appendages. Now isko chhod ke, now let's talk about epidermis itself. Let's talk about epidermis. Now before we talk about epidermis, can anyone tell me what is the function of epidermis? Anyone? Like epidermis bahar ki taraf hai, epidermis is on the outside. So obviously the main function of epidermis is protection. Yes or no? The main function of epidermis is protection. So it is the outermost layer. Epidermis is the outermost layer here. Main helps in protection. It has barrel-shaped cells. Then what is ICS? Intercellular space. Here, if you notice here, in this epidermis of root, shoot, anywhere, leaf also, intercellular space are completely absent here. ICS is completely absent because they are very tightly packed here. Entire epidermis is tightly packed here. Tightly packed. Single layer, it is always a single layer here. Single layer. Okay. Right. Cuticle can be present in the stem. Cuticle will be present in leaf, but in the roots, cuticle is absent. Usse zarurat nahi hai roots ke andar. Right. And okay. Generally, what happens, students? Generally, chloroplast is absent here. Generally, chloroplast is absent here. In the epidermis, chloroplast is absent. Okay, except at a small location which opens up and closes up. Can anyone tell me what is that location on the epidermis? What is that location on the epidermis which generally opens and closes, which helps in the exchange of gases? Students, what do you call that? It is a small opening. It is a small opening on the epidermis of your root, epidermis of leaf, stomata. Very good. So other than the stomata, except stomata, har jagah you will never find chlorophyll. Chlorophyll or chloroplast is only present on the stomata. Okay, so chloroplast is absent except at the guard cells of the stomata. Guard cells of stomata. Okay, so what did we learn right now? If you look at this diagram also, you can understand. Here we can find something called as appendages here. Can all of you notice the appendages? These are called as appendages. It is outermost layer, mainly made for protection. It is single layer here. ICS is absent here. Intercellular space, the space between two cells is not present here. And chloroplast is generally absent here, except the guard cells. That is what we need to know about your entire epidermis. Any doubt, students, in the epidermis? I believe epidermis में कुछ doubt नहीं होना चाहिए क्योंकि it's just the outermost layer. और इस outermost layer के ऊपर से you can see the appendages. The appendages can be trichome, that is your hair-like structure, or root hair. So these two are the appendages. This is the cuticle. Okay, this is the cuticle. Now let's talk more about the entire stomata right now. Okay, now if you look at typical stomata, it has something called as two guard cells. Now before we go to the stomata itself, the diagram on the board right now, is that a stomata of a dicot or monocot? That is very important to understand. अगर आप जो देख रहे हो ना बोर्ड के ऊपर, that is a stomata of a dicot, क्योंकि मोनोकॉट का स्टोमाटा बहुत ही वेरी करता है. मोनोकॉट के अंदर स्टोमाटा जो होता है ना, इन द केस ऑफ मोनोकॉट, इन द केस ऑफ मोनोकॉट्स, जो स्टोमाटा है ना, जो स्टोमाटा, दे आर समथिंग लाइक दिस. In the case of monocots, this is how the stomata looks like in monocots. They are called as dumbbell-shaped monocots. So dumbbell-shaped stomata. So this dumbbell-shaped stomata is found in the case of monocots. And what we generally see in the books, हर एक book में जो दिखा होता है, that is only for dicot one. Okay, dicot is kidney shaped. This is a dicot one. Okay. Now, if you notice in a stomata, it mainly has three parts. कितने पार्ट हैं? तीन ही पार्ट यहाँ पर. सबसे पहला पार्ट है जो दो गार्ड सेल्स जो ओपन होते हैं क्लोज होते हैं. देखे देख रहे हो? These are called as guard cells. ये दिख रहे हैं यहाँ पर. These are called as the guard cells. ये वाले. These are called as the guard cells. These are called as guard cells. These guard cells open and they close. Can anyone tell me how do guard cells open and close? Do we press air inside it? No. So किस तरह से guard cells open होंगे और close होंगे students? The very simple answer is water. Whenever there is osmosis, right? Whenever there is osmosis, whenever the water enters inside, the guard cells become turgid. मतलब फूल जाते हैं guard cells, turgid. So when they become turgid, the stomata opens. अब जब इस guard cell से पानी निकल जाएगा बाहर ना, पानी निकल जाएगा, तब ये सारे के सारे guard cells ऐसे क्लोज हो जाएंगे. This is called as flaccid condition. Flaccid, turgid, turgid, flaccid. So that is mainly controlled by the water regulation. अगर पानी अंदर की तरफ आया तो turgid, बाहर की तरफ निकल गया तो flaccid. Now students, this is the condition, turgid condition. जब turgid condition होता है, turgid, right? Stomata भी open है. Stomata भी क्या है? Open है stomata भी. And you can notice here students, these two are called as the guard cells here, two guard cells. और उसके छोड़ के the whatever we find the middle, right, middle part, that is called as your core or stomatal pore. It is called as what? The middle part is called as the core or stomatal pore generally. Stomatal pore. एक stomata का opening और pore. इसी pore से ना, इसी pore से CO2 बाहर की तरफ जाएगा, oxygen अंदर की तरफ जाएगा, और vice versa also. Because you know plants require both. Plants require CO2 also. Plants also release CO2. They take CO2 also. Okay. Then apart from the students, apart from that, जो ये बाकी सेल्स है बाहर के ना, these are called as epidermal cells. They are called as what students? They are called as epidermal cells. Now sometimes what happens? Some epidermal cells becomes modified. Some epidermal cells undergo changes. They become specialized. Such epidermal cells are called as subsidiary cells. So subsidiary cells are what? Subsidiary cells are basically the epidermal cells only, but they have undergone modification right now. So ये बाहर के दिख रहे हैं जो साइड में अब ये वाले, these are called as subsidiary cells now. They are modified epidermal cells. Modified epidermal cells right now. Okay, subsidiary cells. Clear students? Clear? Any doubt here? No doubt here, right? So stomatal pore, guard cells, subsidiary cells. Now generally what happens is, this is a very important point here, जो इस guard cells के है ना, the inner layer of guard cell is much more thicker. The inner layer of guard cell is much more thicker also. Thicker. Why? Because यहाँ से पानी बाहर निकलता है, water बाहर निकलता है, gas बाहर निकलता है. So this part undergoes a lot of momentum, or lot of exchange of gas happens. To for very same reason, जो अंदर का part है ना, this part is very thick, right? Inner part is much more thicker in nature, जो अंदर का part है, okay, compared to outer part. Now students, here dumbbell shaped. In the case of your monocots, we have dumbbell shaped chloroplast. Okay. Double shaped guard cells, sorry, guard cells. Together all three are called as stomatal apparatus also. Subsidiary cells, stomatal pore और guard cells तीनों मिला के हम कहते हैं stomatal pore कहते हैं इसको. Okay, bean shaped or dumbbell shaped in the case of your monocots. Okay. Now that is the epidermis done, students. Epidermis is done. Now when you look at the ground tissue, what do you find the ground tissue and what exactly is part of ground tissue? Very simple to understand. जो बाहर का है दैट इज एपिडर्मिस, अंदर की तरफ है दैट इज वास्कुलर वास्कुलर टिश्यू। जो बीच का पार्ट है ना, एनीथिंग बिटवीन एपिडर्मिस एंड वास्कुलर बंडल्स, इन दोनों के बीच में जो होता है ना, that becomes the ground tissue, students. For example, this is the epidermis here, then we have vascular bundles here, जो भी बीच का पार्ट है that is called as ground tissue system, that is called as what? Ground tissue system, बीच का है ground tissue system. Okay, now students, now let's understand more about vascular tissue system right now. Now the first one and the second one very simple to understand, very simple to understand. पहला जो है that is called as radial symmetry. Now what exactly happens in case of radial symmetry? In radial symmetry what happens is that xylem and the phloem are present in its own radii, उसका खुद का एक रेडिया यहां पर है। अगर देखोगे आप यहां पर, अब देखो आराम से देखो, if you look at the diagram here, the phloem, xylem, xylem, they are in a alternating fashion and each of them are present in its own radii, उसका खुद का एक रेडिया यहां पर, हर जगह पे उसके खुद का एक रेडिया यहां पर है। Now that is called as radial. Radial. It is generally found in the case of roots along with exarch condition. Now what is conjoint, students? Conjoint is basically a sandwich. क्या है sandwich है यहाँ पर? यहाँ पर हम xylem और phloem को एक साथ रखते हैं in the same radii here. यहाँ पर उनका खुद का रेडियाई था, पर यहाँ पर क्या हो रहा है? xylem और phloem एक साथ है, यहाँ पर xylem और अंदर की तरफ phloem, xylem phloem, xylem phloem, xylem phloem, xylem phloem. This condition is called as conjoint condition. It is mainly found in the case of stem and leaf. So you need to know, आपको एग्जाम की तरफ से याद रखना पड़ेगा, what do you find in roots, what do you find in stem and leaf. अगर ये डिफरेंस आपको पता होगा तो easy. Now what happens, conjoint also has two types. First one is called as bicollateral. इसमें क्या हो रहा है, बाइकोलाटरल के अंदर एक बड़ा और बड़ा सैंडविच बन रहा है, इसके अंदर और बड़ा सैंडविच। यहां पर xylem is present between two phloems here. See, phloem, बीच के अंदर xylem. Okay, so phloem, xylem, that is called bicollateral condition. Bicollateral condition. Okay, then what is collateral condition, students? Collateral condition also very simple. यहाँ पर सैंडविच नहीं हो रहा, पूरा पूरा सैंडविच नहीं, छोटा सैंडविच है यहाँ पर. Here we have xylem and phloem together, that's all. और xylem पता है, xylem अंदर की तरफ है यहाँ पर. If you notice here, xylem is present towards the pith. Pith माने जो center part है ना वहां पर. So you can notice xylem is towards the pith here, towards the pith. Okay, so here we have xylem, phloem, and towards the pith here. Xylem is towards the pith. So that is called as collateral condition, bicollateral condition. Okay, now what happens? In collateral also, we have something called as open and close. Now what is open, students? In the case of open, between xylem and phloem, we find a cambium. That is called as open condition. So xylem और phloem के बीचों बीच there is a cambium here. Now what is cambium, students? Cambium is a tissue which helps in secondary growth. Which helps in secondary growth. It helps in, that is increasing the width of a plant. Okay. Now what about in the case of leaf or in the case of monocot also? Leaf or monocot. Leaf or monocot. In the case of leaf or monocot, we have closed condition. यहाँ पर close हो गया है, यहाँ पर किसी भी तरह का cambium है ही नहीं यहाँ पर. Okay, so between xylem and phloem, we do not find any cambium here. So that is why in the case of leaf and in the case of monocots, we do not find secondary growth. In दोनों के पास we will never find secondary growth here. Never find secondary growth. Secondary growth is always and always and always present in your stem as well as your root. Stem as well as root. Okay, so that is the main difference here, students. But please remember, but in the case of roots, you do not find this. In the case of roots, so technically speaking, we can't write this here. In roots, we can't find this type of arrangement. We do not find conjoint arrangement, never. In the case of roots, we always find the radial arrangement as such. Radial arrangement as such. Okay, so your roots and stem done, students, done. Okay, no. So with that, we are almost done with some questions. So here's the question to all of you. Quickly tell me the answer, students. What is the answer? Stomata and grass are dumbbell shaped, kidney shaped, rectangular shaped, barrel shaped. Stomata in grass are. How are the stomata, students? in the grasses. All of you know grasses are your monocots. अगर monocot के अंदर if it is grass is a monocot, obviously speaking here we will have dumbbell shaped है ना, dumbbell shaped. This question students, what is cortex? This is a neat 2016 question. Cortex is a region found between. Cortex is a region found between. What is cortex? Anyone? See, पहला है बाहर की तरफ है एपिडर्मिस, बाहर की तरफ है एपिडर्मिस है ना, और अंदर की तरफ है वास्कुलर बंडल्स अंदर की तरफ है. And whatever you find in the middle, whatever you find in the middle, that is basically the cortex. So cortex is the middle part, top part is the epidermis, अंदर की तरफ वास्कुलर बंडल, बीच का है cortex, बीच का है cortex. Next question on the board. The specialized epidermal cells, I think आपने आंसर देख लिया होगा. Specialized epidermal cells surrounding the guard cell is called as. I told you students, the other type of epidermal cells is basically the subsidiary cells. Very good. Next question. Vascular bundles in monocots are considered closed because, students, why do we call them closed? Why do we call them closed? Because between xylem and phloem, there is no cambium. There is no cambium here. Cambium is completely absent here. So cambium is absent between xylem and phloem. That is why it's called as closed. What is closed vascular bundle lacks? Closed will lack cambium, obviously. Closed will lack cambium. Chloroplast. So students, that was the basic introduction. That was the basic introduction between what is the outside part, what is the middle part, and what is the end part. इसके आगे आपको कुछ डाउट नहीं होना चाहिए, right? Now I'll teach you the different types of your parts of the entire monocot, dicot stem, monocot root, dicot root, and leaf as such. Okay, बहुत ही easy है students. बहुत ही easy है. Here you have to focus on the diagram mainly. Don't focus on writing much because writing you can understand later on. But I would suggest all of you to focus mainly on the writing or diagrams what is given on the left side. Okay. Now, for example, here is the first diagram to all of you. Okay. Look at the diagram and tell me, student. Forget the writing here. Look at the diagram here. Now, first of all, if you look at this diagram, सबसे बाहर का layer कौन सा है students यहाँ पर? So, which is the outermost layer, all of you? Which is the outermost layer here, students? The outermost layer is the epidermis. Can you see? This is the outermost layer. That is the epidermis. Epidermis is the outermost layer. Done. What does it do? It helps in protection. And in epidermis, do you find any ICS? No, you do not find ICS. How many layers, students? How many layers is it? A single layer. It is single layer. Apart from that, on this epidermis, do you find any appendages here? Yes, we can find root hair here. Root hair. Done. उतना लिखना काफी है उतना. See here. Outermost layer, single layer, root hair is present here, cuticle is absent obviously because it is a root, it is a dicot root. In root you never find cuticle, stomata also you don't find, icis is absent, and here epidermis is also called as epiblema. So outermost layer, protection, single layer, icis is absent here, and it has appendages here. So it's a okay. Now I will not tell you anything, students. Let's understand the diagram and answer. Now, which is the second layer, students? Do you see? Which is the second layer here? Second layer, I see cortex here, students. Cortex is the second layer here. Cortex second layer. Now, if you notice here, cortex is how many layers? You can see many layers here, many layers. Made up of parenchyma. Okay. Then do you find a small space everywhere? Yes, we find small space. So I can tell ICS is present here. Okay. Let's see what do we have here? See. So cortex, right? Here hypodermis is absent. Generally below epidermis, we have hypodermis, but in the case of roots, hypodermis is absent here. Okay. See cortex, multiple or many layers. Parenchyma is thin walled. ICS is less or absent. खत्म, कुछ नहीं हो गया. Right? Very important, very easy to understand. Very easy to understand. Now, tell me, students, which is the innermost layer of cortex do you find here, student? Look at the diagram and tell me. Don't look at the writings, don't look at anything. I will just focus on the diagram mostly here. Okay. Now, if you look at this diagram right now, can anyone tell me which is the innermost layer of cortex here? Innermost layer of cortex. अगर आप देखोगे यहाँ पर, the innermost layer is called as endodermis here. The pink color one is called as endodermis here. Now, how many layers do you find endodermis? All of you. How many layers of endodermis do you find? A single layer? A single layer I can see. Apart from that, do you see any ICS here? No, I don't see any ICS here. नहीं दिख रहा, ICS नहीं दिख रहा. Okay. Single layer, it is the innermost of your cortex and there is no ICS absent here. So देखते हैं क्या लिखा अभी हमने. Is here. Next is called as the endodermis here. Innermost layer of the cortex here. Single layer, ICS is absent. इतना तो easy था. It was easy. Now what we could not tell from the diagram was the shape of the cells here. Now if you look at the shape of the cell here, students, can all of you notice these cells have a very distinctive shape. They are called as what? They are called as barrel shaped cells. This up down up down up down. They are called as what? Barrel shaped cells here. Okay. Now what is very important here is the next point which I will tell you. In the case of your dicot root, endodermis has a separate line here. Endodermis, students, endodermis has something called as what? Endodermis has something called as suberin deposition. Endodermis has what? Suberin deposition. Now what is suberin? Suberin is basically a waxy material which is water impermeable. So it cannot get water, it cannot wet in water. So here in the endodermis of your dicot root, the endodermis has something called as waxy material called as suberin deposition. Now if you look at the deposition, it is present throughout the membrane, radially and also tangentially here. Both the ways it is present. And this when the suberin is present as a strip here, when suberin is present as a strip, it is called as Casparian strip here. Casparian strip. So if I ask you a question right now, Casparian strips are found where and what are they made up of? Casparian strips are made up of something called as a waxy material called as suberin, and they are found in the dicot root. Now what is this? What do I mean by tangentially and radially? I'll show you one more diagram, students. See this. See here, students. See, transverse wall you can find and even radial wall you can find. So throughout the entire endodermis, पूरा का पूरा endodermis के पास on the outside and sideways you can find something called as Casparian strips, and these Casparian strips are made up of suberin deposition. So please remember, this is the characteristics of your dicot root. उसके बाद students, उसके बाद, now after endodermis, which layer do you find, students? Which layer do you find after endodermis? After endodermis, we find something called as pericycle next. Next is pericycle. Okay. After endodermis, next layer is pericycle. Now, if you notice very carefully, students, if you notice very carefully here, very carefully, if you notice, can all notice this is a pericycle here. It is 1, 2, 3. 3 layers of pericycle is there. So pericycle is few layered. Pericycle is few layered it is. Okay. Now it is not made up of green, so you do not write parenchyma here. Let me tell you, student. No. Pericycle. Few layer, students. Okay. Oh, it's a dicot, right? So thick walled, it is made of parenchyma. Now here, what is very important here is the function of pericycle. Pericycle has two main functions. What is it? First of all, first one is initiation of lateral roots. Now, what are these lateral roots? Let me teach you. What is lateral roots? No. If you remember the structure of root, structure of taproot. If you remember taproot, this is the taproot. This is called as the primary root, first of all. Now, if primary root say, you will notice something coming out like this. These are called as secondary roots or lateral roots. Lateral roots. Okay. Now, if you notice here, they are not arising from the surface here. वो surface से नहीं हो arise. If you notice here, they are not arising from the surface. they're actually arising from the inside part here. Inside, you notice, it is not arising from surface. वो अंदर से निकल के बाहर आ रहे हैं. They are endogenous in origin. See, what about root hair, students? अगर यहाँ पर root hair है तो root hair is be present on the outside like this. अगर यहाँ पर root hair है ना, root hair is present like this from outside. Root hair is exogenous. Root hair is exogenous here, but lateral roots are endogenous in origin. देख रहे हैं यहाँ पर, they're arising from the inside here. They're actually arising from the pericycle. इसी पेरीसाइकिल से ना यहां से रूट्स बाहर निकलते हैं, लैटरल रूट्स. यहीं से लैटरल रूट्स बाहर निकलते हैं, यहां से पेरीसाइकिल से इस तरह से बाहर निकलते हैं, इस तरह से लैटरल रूट्स बाहर निकलते हैं. ओके, नाउ फर्स्ट वन इज इनिशिएशन ऑफ लैटरल रूट्स. नाउ व्हाट अबाउट सेकंड वन, स्टूडेंट? सेकंड वन, दैट इज ग्रोथ ऑफ वास्कुलर कैम्बियम ड्यूरिंग सेकेंडरी ग्रोथ. सो इसी पेरीसाइकिल के पास ना, इसी पेरीसाइकिल के पास the vascular cambium also grows. Vascular cambium is part of the secondary lateral meristem, type of your lateral meristem or secondary meristem. This is your secondary meristem, happens, growth also happens from here. And so pericycle is involved in lateral roots as well as secondary growth. दोनों में इसका काम है, इसका दोनों में इसका काम है. Now, so epidermis over, cortex over, endodermis over, pericycle over. Now what about the inside part? Inside we have the vascular bundles here. Okay, now see this, students, they are 2 to 4 in number. 1 2 3 4, यहाँ पर 4 दिख रहे हैं ना, 4 दिख रहे हैं स्टूडेंट्स, देखो यहाँ पर 1 2 3 4, 4 है यहाँ पर, 4 है. So 2 to 4 in number. Okay, and we have exarch condition, students. What is exarch condition? In exarch condition, RSPP, do you remember, students? RXPP from the previous class, all of you remember? From the previous class, all of you remember this? RXPP, yes or no? RXPP, root, exarch, protoxylem, periphery. Protoxylem is present towards periphery here, RXPP. So in the case of roots, we find exarch condition. In the case of roots, we find exarch condition. We also find radial arrangement here. What is radial? Xylem and phloem are present in separate what students? They separate radii. For example, here we can see xylem bundle, phloem, xylem, phloem, xylem, phloem, xylem, phloem. They are present in their own radii. Okay, between xylem and phloem, that is between the xylem and phloem right now, right here, somewhere here, between xylem and phloem, what do you find? You find something called as conjunctive tissue also present. This conjunctive tissue here between xylem and phloem, that can help in formation of cambium ring later on. Help in formation of cambium ring later on. Cambium ring, that is between xylem and phloem, conjunctive tissue is present. Now what is pith? Pith is the middle part here. Pith is the center part here. It is very small, inconspicuous here. Now what is the stele? Stele is nothing but this pericycle on the outside plus the pith and the vascular bundles. All three together is called as stele. So in examination, they can ask you any of the following issues present, any of the following information present on the board. What is the stele? They can ask you. Right? They can ask you what is the stele. You know, they can ask you in dicot root what type of vascular bundles we can find. 2 to 4 exarch and everything. So very easy right now. अगर आपको dicot root समझ आ गया, monocot root is very easy, क्योंकि यहाँ पर epidermis is same, cortex is same, pericycle is same, everything is same to same here, everything is same to same. उसको छोड़ के what about the vascular bundles here? This is the main difference here, students. If you look at the vascular bundles here, let's count students, count करो कितने यहाँ पर 1 2 3 4 5 6 7 8. So more than 6, almost more than 6. Okay, so whenever there is more than 6, it is called as polyarch condition. So in the case of your dicot, 2 to 4, but here it is more than 6, that is called as polyarch condition here. Okay, exarch, same thing, radial, same thing, but it is closed here. Why? Because it is monocot. Monocot के अंदर you will never find cambium whatsoever. So it is completely closed here because cambium is absent here. Okay, here pith is very large, well developed, but in the case of monocot, pith was very small and inconspicuous. Apart from this small difference in the vascular bundles, here, and pith, everything is same to same. Cortex same होगा, epidermis same होगा, appendices same होगा, सब कुछ same होगा in the root and the stem. Okay, in the stem, sorry, in the monocot and dicot, sorry, in the monocot and dicot, not what is it? Monocot and dicot. Okay, monocot is almost the exact copy with small differences. Okay, now let's look at the stem right now, students. Dicot stem, very very simple. Let's see the dicot stem right now. Let's see here. First is the outermost layer here, students. First is the outermost layer here, called as epidermis here. Epidermis. Again, a single layer, main function is protection, everything same. Outermost layer, protection, single layer. You can find trichomes here because it is a stem. Stem has a small hair-like structure. Hair-like structure. So you can find trichome here. No chloroplast whatsoever because epidermis doesn't have chloroplast except the guard cells. Then iris is absent here. Cuticle present or absent. Stomata absent or present. So you can't tell because some plants can have stomata on the stem, some plants do not have stomata on the stem. But stem generally, this is the epidermis here. Now cortex is where it gets interesting here. Okay. यहाँ पर देख रहे हो आप cortex, this entire thing is called as a cortex here. One second. This entire thing is called as cortex here. Cortex. क्योंकि यहाँ से pericycle is starting from here. Pericycle is starting from here. So whatever you find here at this gap, that is called as the cortex. Okay. Here. यहाँ से यहाँ तक cortex. This entire thing is called as cortex. So remember later on. This entire thing is called as the cortex here. Cortex. So if you notice, which is the below epidermis, what do we find? Below epidermis we find hypodermis. Here also we find hypodermis. Hypodermis is made up of collenchyma here. And if you remember, students, collenchyma is absent in monocot. It is absent in root. इसीलिए अभी तक collenchyma कभी आया नहीं था. Now only collenchyma is coming. Make sense? Make sense. Okay. So here we have collenchyma, then parenchyma, then we have the innermost layer of the cortex called as endodermis. So ये तीन layer यहाँ पर है. Let's understand the three different layers here, students. Understand the three different layers here. See here. The ground tissue system, three subzones: the hypodermis, cortical layer, and endodermis. Okay. What is the difference? First of all, hypodermis is made up of collenchyma. ICS is absent here because of the if it is collenchyma, all of you know. If it is collenchyma, the ends, all the ends are what? Ends are connected to each other. They have the pectin deposition also. Yes. So few layer, mechanical support of the plant, photosynthetic also because collenchyma. Okay. Second layer is called as cortical layer, जो यहाँ पर बना हुआ है. The parenchyma one. Parenchyma. This is called as cortical layer. Now what happens in cortical layer? Parenchyma, round thin walled cells, ICS is present here. Normal parenchyma cells. Now endodermis here, students. Endodermis in the case of root, endodermis in the case of stem. Both are important here. Because in the case of root, we had something called as Casparian strips in root. But now we have something called as, in endodermis, we have something called as starch grains. This is here. Starch grain. Okay. So we can find starch grain in the form of a sheath called as starch sheath in endodermis here. So please remember, endodermis in the root, Casparian strips in the stem, there is starch sheath in the stem. Okay. Now students, next one is pericycle. Pericycle is semilunar shape. If you look at here, the pericycle is wavy also. Pericycle is wavy here. Okay. So inner side to endodermis, semilunar, wavy here. Nothing much main function here. Main function, what is it made up of? It is made up of sclerenchyma here. Sclerenchyma. Okay. So brown color you can tell. Sclerenchyma. Then till here, pericycle is over. Now whatever we have here is called as, the entire thing is called as the vascular bundles here. Okay. Now you can find cambium here, so it is open. Okay. Now towards the pith, right? We have protoxylem towards the pith. So RXPP was what, students? RXPP was root, exarch, periphery, protoplasm, protoxylem, students. If the protoxylem is towards the pith now, endar condition. This is called as endar condition, students. Endar condition. And if you notice, xylem and phloem are present together here in same radii. So that is called as conjoint. So conjoint, open, then we have endar condition here. Okay. We can find ring arrangement here. It looks like a ring arrangement here. Okay. Endarc, conjoint, collateral, and open. What is conjoint and collateral, students? Conjoint is they are present together. What is collateral, students? Collateral is basically xylem is present towards the pith. Xylem is present towards the pith, students. Can you see? Xylem is present towards the pith. Conjoint and collateral. Okay. So this you can tell. Open because the cambium is present. Opium open because the cambium is present right now. Cambium is present. That's why it's called as open. So can this root show secondary growth? Yes, it can show secondary growth. Okay. Pith is very large here, very large pith. Ice is present here. It is made of parenchyma. So what I would suggest is learn the notes from the notes. But after that, I would suggest all of you to is in examination try to remember the entire diagram. Like आंख बंद करने से ना पूरा diagram आगे आना चाहिए. Now what is in epidermis, hypodermis, what is present, collenchyma, parenchyma, cortical layer, and then we have the entire endodermis, star sheath. Then pericycle, same will do that shape. Okay, so it has to come in that. Okay, students, I can't speak very loudly because I have cold and cough. Right, if I speak very loudly, my throat starts to hurt. I'm like, today I was supposed to cancel my class, but I still took the class because all of you telling me, sir, I canceled the class. Right, so I'm not able to speak very loudly because I have cold. Like pura gala mera pakad gaya mera gala. Right, it's stuck. Throat is stuck there today. Now, students, now. Now that is between your monocot and dicot stem. Monocot stem is very simple, students. Okay. Sir, I have made a video, students. I have made a video on most important topics of NCGS. Please watch that video. There's a video already done. Okay. Now, monocot stem is very easy. Monocot stem is very, very easy, students. Because epidermis is same. Cuticle, stomata, hypodermis here. But here hypodermis is made up of what? Hypodermis is made up of sclerenchyma. क्यों? क्योंकि यहाँ पर monocot के अंदर collenchyma is absent. Collenchyma is completely absent here. So you will never find here collenchyma here. So we can find sclerenchyma here in the hypodermis is sclerenchyma. Now, what about the ground tissue, students? The ground tissue in the case of monocot is completely scattered here. Completely scattered. You can notice here there is nothing called as your proper hypodermis. You do not have something called as the cortical layer, endodermis, pericycle. कुछ नहीं है यहाँ पर. कुछ नहीं है. Completely scattered. Completely scattered, students. So ground tissue is not differentiated. Parenchyma and ICS. ICS is present here. Obviously, it's present there, students. Okay. Now, what do you see here? Scattered vascular bundles. Done. Now, if you notice here, जो बाहर की तरफ जो vascular bundles है ना, they are more in number, but छोटे 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 students. Okay. In the description, I'll give you the PDF. Okay. जो यहाँ पर दिख रहे हैं आपको छोटे 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 दिख रहे हैं ना बाहर की तरफ वाले. They are called as small vascular bundle. They are more in number, but they are very tiny. But if you notice on the inside, अंदर की तरफ देखोगे ना आप, they are very large but less in number. That is very important here. So outside we have more and small size. Inside we have less but size is more. Okay. Conjoint, collateral, and closed. Conjoint because xylem and phloem are present together here. Collateral because xylem is towards the pith. And closed because cambium is absent. It is a monocot. Cambium is absent there as usual. Okay. Now, here if you notice one more extra thing, students. In monocot stem, phloem parenchyma is absent. This is one, what do I say, distinctive feature of your monocot stem. In monocot stem, phloem parenchyma is absent. Phloem parenchyma, जो phloem में दिखता है ना, phloem, we have sieve tubes, companion cells, right, and phloem fiber. But here phloem fiber is parenchyma is absent here in the case of monocot. Okay, and if you notice here between xylem and phloem there is a separate layer around it called as bundle sheath. That is made of sclerenchyma here. So entire thing is covered by sclerenchyma as bundle sheath here. Water cavities, water containing cavities. If you notice here inside each one of them, if you notice here, you see small dots here. Most of them are water cavities. They can find water cavities inside. So water cavities can be found inside here, inside the ground, inside the entire structure here. Now let's come to the last topic on leaf. Leaf is very simple, students. अगर root stem समझ आ गया तो leaf is very simple. Leaf has two epidermis here. It has upper dermis, lower and upper epidermis and lower upper epidermis. Okay. So it has upper epidermis, or called as ventral side, or ventral side, or abaxial side, adaxial side, and lower side is called as dorsal or abaxial side. Now this is completely reverse of humans. Please, in the case of plants, this is what is true. Okay, so lower side is called as adaxial side. Lower side, lower side is called as dorsal or abaxial back door. So lower back door. This is how I remember it. Lower back door. Lower side, back abaxial, D for dorsal. So lower back door you can remember as this thing. Okay, lower back door. Okay. Now students, if you look at the sectioning here, dicot sectioning students here. Dicot is very peculiar here, students. Dicot is very distinctive in dicot. In dicot leaf, what happens is that the entire epidermis is same. Epidermis is same. No change in epidermis here. But if you look at the cortex here, in the cortex, the entire leaf epidermis is very simple. Upper epidermis, right? Cuticle is present. Cuticle is present because it is a leaf. Cuticle will be present here. Stomata can be present. Stomata is present, obviously. On the lower side, we can find more stomata because it is not exposed to sun. So lower side is not exposed to sun. Upper side is exposed to sun. So वहाँ पर more less stomata. Lower side more stomata here. Okay. But what is important here is the middle part here, students. Middle part. Middle part is made of something called as mesophyll cell. And this mesophyll cell, there are two types of mesophyll cell here. One is called as palisade. This one, देखो, ये वाला, that is called as palisade mesophyll cell. One more is called as spongy mesophyll cell. So in the case of dicot leaf, we have two different types of mesophyll. There is palisade and spongy here, students. Palisade and spongy. Okay. Palisade is arranged parallel to each other. Spongy is present as round oval shaped structures here. Okay. Now students, it is also called dorsiventral leaf. Why? It is called dorsiventral because dorsal side and ventral side. In dorsal side and ventral side, you have two different types of tissues. See, यहाँ पर देखो नीचे की तरफ you can find all the spongy, ऊपर we can find palisade. So different are present, different are present. Okay. Now at the same time, if you look at the monocot, students, monocot leaf is called as isobilateral leaf. Now why is it called as isobilateral, students? Isobilateral. It is called as isobilateral because यहाँ पर ऊपर की तरफ और नीचे की तरफ they have same type of cell here. We do not have palisade, we do not have spongy parenchyma, nothing. Only mesophyll cell here. Only mesophyll cell, students. Only mesophyll cell here. Okay. And stomata are equal on the both sides. Stomata are equal on both sides here. Now what is one thing which is interesting in monocot is that यहाँ पर जो epidermal cells है ना, epidermal cells, some of the epidermal cells, कोई दो तीन या कुछ और epidermal cells, they become large, they become empty, and they become colorless. So there are large empty colorless cells called as bulliform cells. Bulliform cells, and these bulliform cells can take water and hold water. Okay, they can take and hold water, students. They are called as bulliform cells. Bulliform cells, students. Large empty colorless cells. Okay, bulliform cells right now, students. Okay, and obviously the difference in monocot and dicot, all of you know. In dicot, we can find the reticular venation. In dicots, we can find the reticular venation. And in the case of monocot, we can find the parallel venation. Monocot, parallel venation, reticular venation. This difference, all of you know. Okay. So students, with that, today's class is over. Right. In the next class, I will teach you what is secondary growth. Secondary growth in the next class. Next class will be like one small class I'll take. Now I'm not able to talk a lot. Right. My throat is hurting a lot, students. So next class, I'll try to keep it as 20 minutes or 30 minutes. I will send a recording also, which you can all of you can watch. Okay, or I'll make it as a recording itself. That is easy. Secondary growth. Okay. So thank you so much, students. I'll see you next class. This PDF will be available in the description. So go take it down and study well. Okay, students. Bye bye, all of you. Uh, except for Tatwa students, you can always go for Truman or Campbell. Both are really good. But I would suggest starting off with NCERT first. पहले NCERT पढ़ लो एक बार. उसके बाद Tatwa is really good. And apart from that, after reading NCERT, go for Truman or Campbell. Okay.